Murat, I have a question for you because in advance of today's event, we received a number of questions, as you can imagine, around mRNA delivery. We also received a number of people who wanted to hear about specifically RNA. So I wanted to talk to you a little bit about just that. So Cartesian is a biotech focused on RNA engineering, and you have clinical assets in and beyond oncology. So, talk to us about how a cell compares with a nanoparticle in carrying an RNA therapeutic into the body.
Yeah, thanks, Aaron. And Walter, it's just terrific to hear you break it down at that level in terms of the different challenges along the steps of an RNA's journey into a cell when injected either in naked form or in nanoparticle. The way we've tried to overcome those challenges at Cartesian is just really trying to not really tackle them in the first place. And so the way that we sidestep the issue is to, in essence, as Aaron, you alluded to, we use the cell as the delivery vehicle. So we take the cell out of the body for autologous therapies that would be, from the patient himself or herself, or for our off-the-shelf allogeneic therapies, it would be from a healthy individual. And then in our laboratories, in our GMP manufacturing facilities here, we engineer the cell, we transfect the cell with the RNA directly, and then use that cell to infuse the patient, and then use the cell in essence as the delivery vehicle into the body. In the process, we have full control over what happens between the RNA and the cell. We ensure that the cells that we administer, and therefore the RNA therapeutics we administer, meet all the specifications that we set in advance. So, and a part of those specifications ensures that there are a certain, number of cells that are indeed capable of expressing the RNA into the relevant protein and at certain levels, right? So we solved the problem before ever infusing the RNA therapeutic directly into the body. And so the class of therapies that we're developing, we call RNA cell therapy, to distinguish it from conventional RNA therapies where the RNA is either introduced directly or through a nanoparticle. We, in essence, take the cell out of the body, engineer it with one or more different RNAs, and then reintroduce that now RNA engineered cell therapy into the body. And then within the body, since we've instructed the cell through our engineering, that cell goes to where it needs to go, delivers one or more the combinations of RNA therapeutics right at the site of disease. And it has a beginning, a middle, and an end, right? So because we're not permanently modifying the cell with DNA, it has a certain half-life. And so in essence, we're using the cell to sidestep a lot of the issues that Walter so eloquently outlined.